Thanks for joining us. I'm Matt McNulty at Tufts University, Associate Director for our Center for Cellular Agriculture, and on behalf of our Future Foods for Space team, spanning Davis and Tufts, and with TRISH sponsorship, thank you for joining us here for our fourth webinar in our Future Foods for Space webinar series. The topic will be Figures of Merit for Space Bioproduction Systems. I'll give a very quick introduction to the program, just to recap what you might have missed for those who are joining us for your first time. Great to have you, and those coming again. This might be a little bit of a redundant recap, but who couldn't use a refresher these days? And then I'll kick it off to my colleague, Dr. Karen McDonald, for the topic of the day. So to recap, the goal of our Future Foods for Space program is really to prepare a diverse group of faculty, postdocs, and industry researchers to lead in space health, focusing on astronaut food challenges. So this spans the food matrix for nutrition, as well as the health outcomes of food consumption and association, and the logistics of production. You can see our different program elements over in the schematic to the right. And I've already introduced the team. Today, we're focused on the webinar portion of our programming, and we've already had 3 webinars that you can find that have been, the recordings of which and slides of which have been uploaded to both of our project sites associated with Tufts and UC Davis. So these topics include an introduction with the basic concepts of understanding what the food needs are for space missions, opportunities to engage in the knowledge with organizations in Future Foods for Space, and then having real NASA advanced food technology expertise come provide us, their outlook on the current state of the art. And now our last webinar of the series will be, building on understanding the technology into now understanding the costs associated with those technologies. So with that, I would like to introduce our speaker for today's webinar topic, Dr. Karen McDonald. Dr. Karen McDonald is a distinguished professor emerita of chemical engineering at UC Davis, an expert in plant-based bioproduction systems. She has deep experience applying these systems to limited resource environments like space exploration, and we're so excited to have her here today. She's a part of the Future Foods for Space team, as well as several other ongoing projects that we listed in a longer bio as part of the registration. So thank you for joining us, Dr. Karen McDonald, and I'll pass it over to you to take us off from here.
Great, thanks, Matt. And thanks everyone for joining this morning. Let me see if I can share my presentation. And is it in presenter mode or regular mode?
You're good to go.
Great. And can you see my cursor as well, Matt?
Yes, we can see your cursor.
Okay, great. So yeah, I've been asked to talk about some of the work we've been doing on trying to determine figures of merit for space bioproduction systems. And the main goal of this talk is really to give you an overview of the process on how to do this kind of analysis. But to begin with, I think we all understand that food is a mission critical resource. And there was a webinar, I think webinar number 3 with Grace Douglas, which was a great webinar to set the stage for what the requirements really are. And so this slide summarizes some of that. So NASA estimates that astronauts aboard the ISS consume about 2.4 kilograms of food per day per person. And for a 3-year Mars mission, and the focus of my talk will really be on a Mars mission, this would correspond to about 2,600 kilograms of food per astronaut. In addition, food packaging will add additional weight, about 0.36 kilograms of waste packaging per astronaut daily. And the other issue is the long-term stability of food that is brought with the crew. And current targets are to have a 5-year shelf life stability. So that's one aspect that is quite challenging from a technical standpoint. And one of the things in terms of costs is, how much mass is required to launch 1 kilogram of payload. Um, this is often now referred to as a gear ratio, but a recent report indicated down here, the 2023 Moon to Mars Architecture Concept Review, talks about some of the mass challenges for a round trip Mars mission. And in that, they estimate that about 10.6 kilograms of total launch mass is required for 1 kilogram of payload, and this is for a very specific mission architecture. That is a 1,100-day, 4-crew round-trip Mars mission. This number can change dramatically as that mission architecture changes, but for now, let's just use that number. And if we use that number, then we can calculate that about 110,000 kilograms of mass is needed just for the food if we brought all the food for the crew for the entire duration of the mission. So that's a lot of additional launch mass. And so some of the options, and these are some of the options that some of the seed projects through the TRISH Future Foods for Space have been working on, and that is developing high-density nutrient foods with minimal packaging, or edible packaging, for example, or creating food systems with longer shelf life. Some other options are using local Mars resources where possible, or implementing advanced recycling systems, particularly for the packaging waste, or growing and producing some food during transit and on the surface of Mars. Now, these last three could be considered sort of bioproduction strategies, and it's important to realize that they'll have their own requirements. That is, they'll need specific equipment, they will need particular inputs and generate certain waste. And so they have costs associated with them as well. So that's sort of the goal of a figures of merit analysis. So what are figures of merit? And I don't know if Matt said, but if you have any questions, just enter it in the Q&A, and then at the end of the presentation, we can address those questions. And Matt will be looking at them as we go along. What are figures of merit, or FOM? On Earth, we might consider the costs of food production in dollars, in economic terms, such as capital costs, what's the equipment needed to produce the food that's going to be used, how much does the facility cost in dollars, usually millions of dollars, or in operating manufacturing costs. That is, how much does it cost in material inputs, consumables, labor, energy, waste disposal, everything like that, to make a kilogram of product. And that's usually in dollars per year or dollars per batch. But for space, these costs are usually in terms of these figures of merit, and that includes the mass, because as I already mentioned, due to these gear ratios, the mass payload plays an important role in how much mass is needed to launch and get all the way back, get to Mars and back. Volume is really at a premium. There's not much space, as you can, if you've seen any of the capsules that the astronauts are in, or even the International Space Station, volume area are at a premium. Power, it's not readily available. You have to make certain you have the power needed to run equipment. And a really important one is crew time, because the crew has lots of different demands for their time. And so if they spend their time in a bioproduction process, making a food, for example, that takes away from the time they can do other things. But note, in addition to mass, volume, power, and crew time, there are many other considerations. For example, how long does it take to make a kilogram of food, particularly if you're in a situation where you don't have enough food? How safe is the product? How safe is the process? How reliable is the process? And what is the nutritional content of the food? So those are other considerations, but for now, we'll just focus on these FOMs. And in 2024, TRISH issued a request for proposals called the Figures of Merit Analysis, How to Get Biology Engineered for Exploration of Space, or FOMBs. FOMBs is what we refer to it. And we were really fortunate to get one of these grants, and I'd like to thank TRISH specifically for giving us this opportunity to take our work and put it in the context of an FOM. And these are really analyses to implement, to analyze the cost to implement new technologies in space for food production or supplemental nutrients or pharmaceuticals. So that was sort of the goal of the FOMBs project. So in our project, in our FOMBs project, our objectives really are to quantify the required mass, volume, power, and crew time required to produce a weekly dose of a therapeutic. So we were more in the pharmaceutical side, but some of what I'll present, or at least the framework, will apply for food production systems as well. And our therapeutic was a parathyroid hormone Fc fusion protein, and this is a protein that we've been working on in our group to make in a variety of different platforms, plant-based platforms. And so we're going to compare these different plant-based production scenarios. So I'll just give you an example of 3 of the different platforms you might consider. You might consider flying the drug. It's already approved by the FDA, or at least the parathyroid hormone itself is a drug right now called Forteo for osteoporosis. You could fly that. You'd have to store it under refrigerated conditions and have enough for daily injections for the entire crew. But another option is to use transgenic lettuce that has constitutively or continuously produced the PTHFC in the plant tissue. And this is something that one of our students, Shanice Taylor, has been working on. And the idea here would be that you would just bring seeds with you, you would grow lettuce, that you probably have the facilities, plant growth facilities to grow things like lettuce. But we'll include those in our FOM analysis as well. And then potentially you could deliver it orally. Another option would be to transiently produce the PTHFC in lettuce using a gene gun strategy. And this was actually a experimental project that Trish had funded us as well before this project started, to get the experimental data, not in lettuce and not with PTHFC, but for other proteins. And in addition to those 3 platforms, we are also looking at 3 other platforms, which I won't talk about today, using Nicotiana benthamiana. So one would be transgenic Nicotiana benthamiana, not an edible crop, it's tobacco. And another is transient expression using agroinfiltration. And the third would be using transient expression using a viral vector. I won't talk about those today. I don't have enough time, but you can see that there are oftentimes there are many alternative strategies to produce something, and having a way to analyze the figures of merit and the costs associated with it are important. So just to talk about the general approach that we took, and is one that I think is a valuable approach. First, obviously, is to define the scope. What is it that you're making? How much of it do you need? And general modeling assumptions, and then scenario-specific assumptions. So, for example, in this case, you have to grow the lettuce. Obviously, there are costs associated with doing that. But over here, and this is work that's done by Katie Haddad, over here, you take that lettuce then and you bombard it with these basically projectiles, gold particles coated with the DNA that contains the expression system for the protein you're trying to make. So there are some advantages for that because you can work with wild type lettuce. You don't have to have transgenic lettuce. You can work with different kinds of plants. So it's a little bit more flexible for, it offers more flexibility in the bioproduction, but there are scenario-specific assumptions here, but there's some common assumptions as well. And then the next step is to develop block flow diagrams for each of the production scenarios. And in those block flow diagrams, and I'll take you through a couple different ones, one for the transgenic lettuce and one for the transient production. Show you how the block diagrams are constructed, but it's important to identify what are the system components needed, what are the inputs for the production process, what are the waste outputs, and what are the useful outputs. So, for example, the roots of the lettuce might be a waste output. It's an inedible part of the plant. On the other hand, a useful output might be the oxygen that's generated during the growth of the plants. That might be a useful product. The water that's generated through plant transpiration, things like that. And then the next step is to use experimental data or published data or existing models to do material balances so that you know exactly how much of the inputs do you need to generate the desired amount of output, which in this case is parathyroid hormone, FC fusion protein. And then you need to, once you've done that, you need to quantify those metrics, the mass, volume, power, and crew time as a function of the amount of dose needed per crew member per week. So some of the deliverables that we are generating are basically Excel spreadsheets containing these block flow diagrams and the figures of merit, along with the assumptions. These are models. Models are always built on assumptions, and clearly listing the assumptions and the source of the information and data used in the model are really important. But what is the potential impact? It can really assist in decision-making to try to identify the most promising space-relevant bioproduction strategies. In this case, a therapeutic production strategy, but it could be food, it could be a material. And then it also helps you identify critical research needs to minimize the impact. And you'll see as we go through our FOMBIs, there's some scenarios where it's just impractical using the current system. And then the FOMBIs is also relevant to even therapeutics production in low resource environment on Earth, because you might want to know how much power is required or how much mass is needed. So just a little bit of overview of our target protein. It's a parathyroid hormone FC fusion protein. Parathyroid hormone is used for bone regeneration. So Forteo is an FDA-approved drug, which is just the PTH amino acids 1 to 34. It's for osteoporosis administered through injection. But it stimulates bone regeneration by activating osteoblast cells by binding through the PTH receptor on cell surfaces. We've linked it through a peptide linker to the FC, or crystallizable fragment antibody domain, from human IgG1. And this is really to enhance the half-life of the protein in the body and hopefully reduce the number of doses needed per time, so to reduce the dosing frequency. Just some technical things. It's designed to be aglycosylated and retained in the ER. And in addition to the injectable form of PTH, there's also an oral form that's in human clinical trials called EBP-05. And so there is the potential for oral delivery of this hormone. So what are our general modeling assumptions? First of all, in this analysis, we're only modeling the upstream or production part of the process. That is up to the point where we've got PTHFC made. We're not considering downstream processing if needed in this analysis, but it can be done. And there are some other nice papers like this paper here that came out in 2023. Is on theoretical design to produce recombinant proteins and focuses on a protein, an enzyme needed for CO2 scrubbing and made in E. coli, but they focus a lot on the downstream processing. And even Matt has published a really nice paper on ESM or figures of merit for pharmaceutical purification, which, yeah, I should have added that too, but yeah, it's a really nice analysis of an alternative downstream processing, but this doesn't consider that part, just to let you know, this is part of the scoping. We consider 600-day Mars surface operation. And then we also consider that we're going to make weekly production batches of a dose of PTHFC. We picked 600 days. That was also the assumption in this paper below as well. One thing that we're not so clear about is what is the dosage level. So we investigated weekly doses of PTHFC monomer per crew member per week over a fairly wide range, going from 0.1 milligrams per week all the way to 100 milligrams per week. And we thought this would be useful as well, because if you use less to make some other drug, if you know what the FOM is as a function of the amount of drug needed per week, you can translate the FOMBI's, the FOM analysis to that scenario as well. But just to let you know, Fortio is 20 micrograms per day. So over a week, it's sort of in this low range here, 0.1 milligrams per crew member per week. And the more experimental clinical trial drug that's orally delivered is it a higher range, and that's more like over here to the 10 to 20 milligrams per week. So just to cover the entire range, we looked at a broad spectrum of dosage. I'll focus today on plant production systems that utilize lettuce. But we have done some work with tobacco, and you could extend this analysis to spinach or other crops as well. So there's a lot of opportunity to expand the model. We are neglecting a number of things. We're neglecting analytical equipment, for example, to measure PTHFC dose. Obviously, that would be needed. And we've also neglected on-planet seed production. So we assume that the lettuce, we bring all the seeds necessary for the lettuce, but obviously you could have the lettuce make seeds and then collect those and use those for the next batch to minimize the seed amount, the mass of seed needed for the. So this is sort of a typical mission scenario. This one is, maybe 967 days total, with 600 days on the surface of Mars and 87 days to transit to Mars, 180 days transit back. This 600 days corresponds to 86 weeks, and so we'll assume that we need to design our system, our bioproduction system, to make 86 weekly doses of PTHFC. We're not considering making any of it in transit, either to Mars or back to Earth. But basically, you look at each unit procedure. So for example, the production of lettuce might be one unit procedure. There might be a lot of operations required in that unit procedure. You might have to seed, provide new seeds, new plantings, germinate those, grow the plants. You have to harvest the plants. And for each of those operations, you'll have maybe a labor cost, a crew time cost for that operation. You'll have equipment that is needed for that unit procedure. This is more in the systems level. and that equipment will have mass, volume, and power. This equipment might be, for example, the hydroponic growth chamber that the lettuce is growing in. You might have raw materials like seeds that you have to bring, the nutrient mix, you have to provide water. And then in terms of crew time, you'll have crew time to harvest the leaves. And you'll have power as part of the requirement for the lighting systems for the plants. We're assuming the plants are grown using LEDs as a light source and in a hydroponic nutrient solution. So let's go on to the actual models. So the transgenic model, and this was work that really is done by Shanice, who's actually in the backstage, I think. So this is the transgenic model assumption. One assumption is the lettuce plants are harvested after 28 days after seeding. These are transgenic lettuce plants. They already contain the PTHFC, but they're harvested after 28 days after seeding. That means at any given time, you have to have 4 batches of plants available. And this number is one that could be reduced. In fact, some of NASA's work has shown that this could be less than 28 days, and that will obviously minimize the area needed for plant growth, and yeah, it should make the cost a little bit lower. The production level in transgenic lettuce at day 28 is assumed to be 7.1 milligrams of PTHFC per kilogram fresh weight of plant tissue. And this is from some of the experimental work that Shanice has done. But I would have to say this is at the low end, but I'd also say that this is a really critical parameter in the model, because obviously if this were 500 milligrams per kilogram fresh weight, everything shrinks in size, the size of your growth chamber required, and just about everything. So just keep that in mind. We are planning to look at varying, this is often called the titer or content, protein, recombinant protein content. But knowing what this is really critical. The lettuce plant is, each plant is assumed to be 192 grams fresh weight at harvest. Some of these numbers come from a document that NASA produced called BVAD. I'll introduce it a little bit later. And the area required for one lettuce plant is 0.052 meters squared, so you can have about 19 plants per square meter. And as I mentioned, we're neglecting analytical equipment and seed production. So we took a lot of the assumptions in this model from NASA's Life Support Baseline Values and Assumptions document, BVAD from 2022, and specifications like for the growth chamber, size, square meter, and volume. We took those from a certain table in BVAD. And for lighting, BVAD used high-pressure sodium lamps, and we switched over to LEDs. And this was based on some work that we've done with Bruce Bugbee at Utah State University and Paul Kusuma, who were part of our NASA Cube Center. And they've done a lot of work on lighting systems for plant growth. So they helped us with the design of the lighting system, LED aluminum lighting panels. We assumed red and white LEDs, and they gave us some of the metrics. Yeah, what's the mass of one LED panel? What's the volume? How much power does it consume? And what is the photon output from that system? So additional system inputs and outputs, we had a freezer so that we could store the plant tissue once it was harvested. We have harvesting tools, collection containers. We had to provide the seeds, of course. And then for the plant growth, the composition of the plant and also the nutrient solution, BVAD provided that information as well. And we also took into consideration that if we eat these lettuce plants that have the PTHFC, they're not only providing the PTHFC, but they're providing calories, macronutrients, micronutrients, a lot of other useful outputs. And so, as I mentioned, we start off with a block diagram. And the block diagram is important. The blocks actually represent the system or infrastructure components. So you might have, in this case, you have really just two steps: plant growth and plant harvest. For the plant growth, you have your plant growth system, that is the hydroponic system that you're going to be growing your lettuce in, including the lighting fixtures. And then over here on leaf harvest, the system or infrastructure or equipment that you will need includes your harvesting tools, your harvest container, and the freezer that you're going to store your tissue in. And you'll have inputs. Here, the inputs would be lettuce seeds, water, plant nutrients, carbon dioxide, and another input to the plant growth would be photons, and these will be generated from the growth chamber, so they're not an external input. They're actually part of the lighting system, but they are needed, obviously, for growing your lettuce, and that will generate plants that then can be harvested. And the outputs would be from plant growth, you get some useful outputs. You get oxygen production and you get water through transpiration. And during harvesting, you're going to get useful outputs as well. You're going to get the PTHFC, you're going to get the plant biomass that provides calories, macronutrients, micronutrients, and water. And then you'll generate some waste as well. In this case, it'll be inedible biomass. Oops. So with those assumptions, you can basically we set up spreadsheets to track all of the figures of merit, and these include let's see, we've got the this is transgenic lettuce figures of merit for a particular range of doses from 0.1 to 1 milligrams of PTHFC per crew member per week. And what I'm showing here are the mass figures of merit, volume, power, and crew time. And this is in units, mass is in units of kilograms per crew member for the entire mission. Volume is similar, cubic meters per crew member per mission, power is kilowatts per crew member per mission, and crew time is crew member hours per crew member per mission duration, the 600 day. And for each of the doses, so this is 0.1 milligram dose, 0.2, and 1, you have the system mass, for example. And for the system mass, it's mainly the plant growth chamber. I know the colors there's so many different things, the colors are a little bit hard to see, but the plant growth chamber is the main mass that's needed for this particular process. As far as inputs, water dominates the inputs, but transpired water also dominates the outputs. So in a way, if you can recycle the water that's generated through transpiration, you're going to really save a lot of input mass. So that this is one of the outcomes of doing this analysis. It kind of points to what you might try to recycle and what you don't have to worry about recycling. In this case, waste is pretty negligible because the waste is just the inedible biomass, which is pretty small fraction of the total biomass. And then let's see, if you look at volume, again, the plant growth chamber dominates the main volume needed. You have the volume of water, but you also have your gases, carbon dioxide and oxygen as an output. In terms of power, it's kind of split between the power needed for the plant growth chamber and for the lighting system. But pretty constant. You'll notice that for this range, everything is fairly constant. There's not a lot of change. For crew time, even though the color here indicates really carbon dioxide, it's really the operation of growing the plants. So that's mainly what the crew is working on, growing the plants and harvesting the plants. Um, but if you go from 2 to 10 milligrams of PTHFC per crew member per week, you start to see more of an increase with dose. So the trends are the same. You still have the same major contributors: water input, water's output, and the plant growth chambers, the system mass. So all of the trends are pretty similar, but you see dramatic increases in the figures of merit. Obviously, because you you would think that you would see this because you have to have more area, because you need to have more plants, because we've fixed the expression level or amount of PTHFC per kilogram of fresh weight of lettuce. Um, and then you can go from 10 to 100, and again, these numbers get larger, the scales are bigger, but the trends are pretty much the same. Um, so it's once you set up your spreadsheet, you can make changes in things like the dose or the even the titer, how much PTHFC you have in the plant mass. So I'll move on to the gene gun assumptions just to show you how things can get a little bit more complicated, and the cost of complexity, I guess, is one of the things I'll point out. So this is a plant production system that the good thing is it uses wild type lettuce. So you don't have to have special seeds that you may want to keep separated from your regular lettuce that you're eating. And so that's nice. You can just use the wild type lettuce. We assume that the growth of wild type lettuce is the same as the growth of the transgenic lettuce. So the lettuce plants are also harvested at 28 days after seeding. You'll have 4 batches of plants at any time. The production level was assumed to be 2.5 milligrams of PTHFC per kilogram fresh weight of plant tissue, so lower than the transgenic lettuce. I still think the transgenic lettuce, we can get that to be quite a bit higher, but and also that one shot of using the gene gun, you produce 0.3 micrograms of PTHFC. The same assumptions are used for the plant weight at harvest and the area required for one lettuce plant. So those are all kept the same. But some additional equipment is needed, this particle bombardment system, and here we assume we use the Biorad Helios gene gun. It takes batteries, so it can only do 1,000 shots per battery, so you have to bring batteries with us. That's part of the cost. And the other thing in what we assumed was that the bullets would be stored desiccated in cold storage under refrigerated conditions. This turns out to add a lot of cost, and it's obviously one thing that I think we really need to determine is 4 degrees C storage of these bullets, which are it's just a plastic tube coated in the inside with your gold particles that are coated with the DNA. If it can be done at room temperature, it's going to save a lot of cost. But just to let you know, we assumed that we needed cold storage for that, and that all consumables for the gene gun would be packed at launch. So the block diagram looks a little bit more complex for this system. Same inputs for growing the plants and same plant growth system, but the additional part is this particle bombardment. You need the gene gun system, this little gene gun. You need a gas cylinder. It uses, in this case, helium, but any inert gas could be used. That's what provides the blast that blasts these particles into the plant tissue. you need gas regulator, you need Ioneer protection, and bullet cold storage is sort of the biggie. And so you need to bring the bullets with you and bring batteries with you, and those are sort of consumables. And then you'll generate some waste, and you will also generate the inedible lettuce waste. And then you have the similar outputs as we had before. So if you look at the gene gun figures of merit, and again, it's laid out in the same way: mass, volume, power, and crew time. In this case, 0.1 to 1 milligram. The first thing you'll notice, the scales are much larger. And if you look at the system, this little orange part here is the plant growth chamber, but you have this part, this contributor here, which is the refrigerators needed for the bullets. So this is what I mentioned: if you can if you can store those bullets desiccated just at room temperature, you can greatly reduce your mass requirement. But the water is very similar. Both of these are water. We just haven't colored the water output differently as Shanice did, but pretty similar. And for the volume, yes, the volume of the refrigerator also plays is a contributor. And the power, the refrigerators play a big role, and crew time is much higher, assuming, and this is assuming that there's a crew member that is actually doing the particle bombardment on the plant. And obviously that's something that can be automated. And if it was, then you would greatly reduce this crew time. In fact, some of the crew times when you get to the really high dosage, so if I'll just skip through the 2 to 10, it's pretty similar, 10 to 20. I calculated that this is corresponds to 26 hours of crew member time per day, which is totally unreasonable. You won't have a crew member that's sitting there for the entire day doing particle bombardment, and there's not, it's 26 hours in the day. So it really can point out some things about scaling of these systems and constraints that you will be hitting when you start to scale. And either this could mean that you have to obviously work with pharmaceuticals that are lower dosage, so that you can accommodate that, or be able to achieve higher titers, higher amounts of PTHFC per fresh weight. But an additional consideration is that these are nice figures of merit, and this is actually what Trish really wants to see. And one of the nice things about separating these out is it allows you to make choices that may be based on, maybe you have you're in a scenario where you've got a lot of power, so you can maybe neglect this and focus on the mass. Or maybe you are really restricted for volume, and so you want to make decisions based on the volumes required for the production system. Or maybe crew time is really the essential thing. So it allows you to make decisions based on the particular situation you're in. But if you want to do things like, let's compare the gene gun to the transgenic lettuce, then we move to a single metric, often called the equivalent system mass metric, that combines those mass, power, volume, and crew time into a single metric that we can now compare alternative production strategies with a single metric. But I just want to mention that there are a lot of additional metrics that you might want to include other than just mass, volume, time, and crew time and power. You might want to include some of these metrics as well, which I don't have time to go into, but there's a nice paper that came out of our CUBES team that Aaron Berliner headed up that sort of describes some of these other metrics that might be important. But let's look at equivalent system mass. And so this is in units of equivalent kilograms. And this is a equation that we sort of adapted from the paper that I mentioned earlier. We just broke it up into, this is a summation over S stands for system components. These would be like equipment. I is inputs. OW is the waste outputs, and OU is useful outputs. So it's a summation of all of those things in the block flow diagram. The top part is the system. And so you have the mass of the system that doesn't have a equivalence factor associated with it. It's just the mass of the system. But when you go to volume of the system, you need to multiply by a volume equivalency factor that you can get from, for example, this table here, 2022 equivalency factors for shielded volume. It would be 79.3 kilograms equivalent per cubic meter. And you have a similar factor for power, for thermal cooling, and for crew time. So that's for the system. And then for your inputs and outputs, that depends on how many batches you have per year. So that's one thing we haven't varied, is how many batches per year. But that could be varied as well. Instead of a week, maybe every 2 weeks, or maybe every day. So these are the process inputs. Again, very similar waste outputs and useful process outputs. And I put a negative here because if it's a useful output, like oxygen or in this case, calories, it's things, those are things that you didn't have to fly up. So you should get a credit for a process that provides a useful process output that means that you don't have to fly that equivalent mass up. But just to show you what the equivalent system masses look like for transgenic lettuce, is shown here. It's mostly system, input ESM, and output ESM. Very little waste, so the system doesn't generate much waste. The total ESM is listed over here in this table if you want to look at that. So that's, and it also kind of tells you at what point do things start growing exponentially. They're growing significantly, and maybe where you might need to operate in a dosage level to be in a reasonable equivalent system mass. The gene gun, these numbers are way high, much larger, although they do start out small as well. But again, it's important to look at the trends are pretty similar, though. It is sort of the growth here, and these are the total ESMs. So given the time, I just want to give some suggestions for doing a FOM analysis. One is start by defining the scope. And there may be things that you want to vary, in our case, dosage, and in the future, titer of our PTHFC, the general modeling assumptions. You develop block flow diagrams, being careful to identify all of the system components, every piece of equipment that's needed, every input, every waste output, every useful output. But separate the system component into infrastructure. And the reason that that's important is that equipment is used for multiple batches. And that's why doing this on a per batch, like if you just looked at the FOM for a batch, and you said the growth system is going to be needed just for one batch, it's not going to be reasonable, because that growth system is going to be used for every single batch. Separate the waste outputs from useful outputs so you can get credit for the useful outputs. Separate the in situ process inputs from flown inputs. We assumed all of our inputs were flown, maybe except for water. And you have to do mass and energy balances. I think in any of these models, they're really only useful if you list your model assumption, your parameters, and the sources of those parameters, because they change with time. Sometimes, like if you look at the BVAB from 2018 to 2022, some of those parameters have changed. But the ESM can be a useful metric to compare alternative production schemes, but will depend on mission architecture. So that is, how close are the planets when you fly to Mars? So how long is that duration? How long you're staying on the surface, and how long does it take to get back? So I just want to acknowledge Shanice, who did all of the work on the transgenic lettuce and the growth of the lettuce, and then Katie, who did all of the modeling work on the gene gun, um, and also some folks that provided us really with useful information that became part of the model. And definitely want to thank Trish for funding this FOMBIES project, as well as funding the the ship, Future Foods for Space, and also NASA and our CUBES team. And with that, thanks for your attention, and be happy to answer any questions you have.
Thank you so much, Dr. McDonald. That was a fantastic talk. Learned a lot. If okay, I'll take this opportunity to kick off the questions myself. Um, and this is one of those times where there's something you said very early on that sort of stuck in my head, and I couldn't quite get it out. But you were talking about how figures of merit analysis, typically used for these space missions and architectures around them, that they could be useful for limited resource environments, other types of limited resource environments. Um, in my admittedly somewhat limited experience with looking at Department of Defense and other sort of defense applications, or humanitarian efforts terrestrially, I haven't seen much of looking at these sort of figures of merits, looking at the mass volume and creating sort of economics around that. Is that something that you've seen popping up, or there being any discussions around it? Because it's a really interesting thought.
Yeah, no, I haven't seen it either. I think you're absolutely right. Everyone focuses on cost of goods sold,
Hmm.
um, which is somewhat related in some respects, at least from the standpoint of, power costs money, and if it's something that requires a lot of energy, it's going to be more costly. But yeah, I haven't seen it couched in for a given production system. But I think it's important, especially for field deployable. I know folks like DARPA are interested in, yeah, how small can you put a food production system? What's the footprint? So that corresponds to the volume needed, or what's the mass of the production system. So I agree with you. That's a great observation, and one that, yeah, could start applying this to that.
Yeah, yeah. Thanks for seeing that idea, and thank you. So I'll move to the chat right here with the next question, and it's really about refrigeration issue, and it's somebody who is working on a national grant project in India that's called CryoServa, looking at portable storage system for off-grid low temperature storage. Wondering about how there could be collaboration across these environments, and maybe even, looking to collaborate specifically with you. But if you were to give some advice on how one could start to think across these spaces with the cold storage and the gene gun, could you speak to that a little bit?
Yeah, definitely. I mean, our goal with the gene gun would probably be to do experiments to see if it performs similarly if you just, have it desiccated in a container at room temperature versus refrigeration. Because I think if you can get away from refrigeration, that would definitely be the best. But there are going to be many bioproduction systems that require refrigeration, especially for food systems, but also maybe for other kinds of systems. So I think the work that that Susan is doing would be really valuable. If you look at the BVAD, Susan, there's a lot of pages, many pages, dedicated to refrigeration systems. And so I think it's an important uh, problem for NASA and for exploration, because no doubt there are going to be components or products that will require refrigeration. And one of the things I didn't show you was the flown system, but mainly much of the, like you're flying these injectable pens with your drug, right? And those have to be refrigerated. It's critical there that they're refrigerated over 600 days, so you have to have enough storage, refrigeration storage to take enough pens for 600 days for number of crew members. So it's a lot of pens. So you need a lot of refrigeration, and it's the main contributor in that case is the refrigeration. So yeah, happy to talk to you more about it. And yeah, I think hopefully my email is available or was on my slides. I'm not sure.
We'll make sure that that's available. Yep, when we send out info after the webinar. Thank you, Karen. So another question that came in was really about recycling, and I think you showed the massive amounts of water and the impact that could happen there. When it comes to other components for recycling, I think, for example, the question asked here was about urine recapture of bio compounds within these systems. You know, how are you able to approach those and decide what you can or cannot include? And then, specifically with urine interaction with the bioproduction systems, how is that being approached?
Yeah, we haven't worried about that too much yet, but there's a lot in BVAD about urine capture or water recycling from urine. But there's also the nitrogen availability in urine that could be used to supplement some of the nitrogen needs of plants. Um, and I guess the question there is you really do have to develop the processing system to take that raw urine and get it into a form that you could apply it to the plants. And I'm not sure what that system looks like, but from a FOMBI's analysis, you just need to make sure that you design it and have it tested and include all the inputs, outputs, and equipment associated with that process.
Thank you. And I might take this last minute to ask one more question. So it's really about the difference between cost analysis for terrestrial versus space bioproduction. Terrestrial, you have a lot of mature systems that have, scaled up successfully. And so you have these really nice anchor points on costs that you can rely on, but you don't have nearly as many of those. You were referencing a couple times NASA's BVAD document, and then even, talking about how there are some assumptions there, like with lighting, how you sort of move to LED where they had started with the sodium. But even outside of that, with this uncertainty, how does that approach how you design your analysis or the questions that you're asking right now?
Yeah, that's a great question because I mean, one of our challenges is we come up with these ESM numbers, and what do they mean? Are they high? Are they low? Are they because there haven't been many, well, there haven't been any bioproduction system on Mars as far as I know of. But I would say the way to start would maybe to be doing analyses that can be validated on the ISS, because even though I considered Mars because that was the context that FOMBI's was about. I think looking at like the moon or ISS and then validating these equivalent system mass numbers that are generated, it would be one of the ways to go. Um,
Hmm.
yeah, and ISS might be the easiest thing to do. I mean, there are experiments done on the ISS, and some of them involve bioproduction to some extent. So it may be that you could take some of that data and generate a FOM analysis and then compare with, yeah, what was actually required in terms of mass, volume,
Hmm.
power, crew time.
Yeah, that makes a lot of sense. And I hope to see the community gathering around some of those proving ground options. We did have a question come in just now. I don't know if you have to leave this moment or if you're interested in fielding one more, Karen.
Sure.
But we have a question here from, it looks like perhaps preferred name Dove. I'm just going to read this one directly. Given the limitations of conventional gene delivery and microgravity, how feasible is the use of bialistic methods like a gene gun for functional genomic studies in space, in space bioproduction systems, and what challenges remain for efficiency and viability?
Yeah, thanks, Barbara. There's a lot of challenges and efficiencies, certainly. That's one of the reasons that we've had so many bullets required and such a high refrigeration. And yeah, efficiency is a key issue. That's a big challenge. Um, in terms of the microgravity issue, so we're assuming we're on Mars, so it has 3/8 of the gravity of Earth. So there is gravity on Mars. So that's we haven't considered microgravity, but I agree. I don't know if it would work. I don't think I would be shooting the gene gun around the International Space Station, but there are other ways for gene delivery. There are a lot of, like, even agrobacterium involves vacuum infiltration, so you use a vacuum basically to suck the air out of leaves and replace it with the agrobacteria. So that might be implementable in microgravity. But I think we do need to think about gene delivery techniques and approaches, and there are lots of different ones. One of the other ones we're using is just a engineered plant virion that we can mechanically wound the plant tissue with. So that could work. But there are pros and cons of every system that you can think of. But yeah, there are coated nanoparticles that could be used using sort of a vacuum approach as well. So, but yeah, I'm not thinking that biolistics would be used in transit or in the International Space Station, but
Great. Well, thank you so much, Dr. McDonald, for your time on this really important topic. I know I appreciate it, and I'm positive that our webinar participants did as well. Webinar participants, thank you for showing up. We appreciate your participation. You can always feel free to reach out to us afterwards. We'll send out contact information along with the webinar recording when we've got that all situated. With that, we'll bring to a close our 4th webinar. Thank you all. Have a nice
yeah,
day.
thank you, Matt. Thanks everyone for attending.
Yep, take care.